All right. Well, welcome to our second seminar for the Midwest Bioinformatics Showcase Series. This series is being organized by myself, Haley Carter, and Arun Seetharam. I'm at Northwestern University in Research Computing and Data Services, and Arun is at the Rosen Center for Advanced Computing at Purdue. We are organizing this partially just to highlight all the really cool science that's happening at both institutions, and to give graduate students and postdocs another avenue to showcase their work. The focus is on work that either incorporates or advances the fields of bioinformatics and computational genomics. And if you in the audience are doing any of this type of work at either institution and are ever looking for more help, either in finding the appropriate computational resources or troubleshooting scripts or needing someone else to take a look at either your workflow or experimental design or resources or anything like that, please do reach out. We're happy to help facilitate more of this type of science, either to myself or Arun or either of the groups, the larger teams that we work on. So today's speaker is, oh, let me just make sure I can see my notes before I start here. Uh, it's my pleasure to introduce today's speaker, Yoon, a PhD candidate in the Dawoodi Lab at Purdue University. His research uses whole genome resequencing and landscape pangenomics to study genomic erosion, local adaptation, and climate vulnerability in species of conservation concern. He has worked with many different groups of taxa, amphibians, fish, mammals, and lately mostly birds, I believe, though I'm not quite sure what organism or organisms we'll be hearing about today. Jung Yoon is defending his PhD imminently, I believe, and will be joining the Edwards Lab at Harvard University shortly as an environmental fellow. He is also the author of an award-winning book about his experience in the Peruvian rainforest. So if you are a reader of Korean, you can check that out too. Okay, without further ado, please take it away, and I will hand over the screen share to you.
Thank you for a fascinating introduction. I believe my screen is shared now. Can you see my screen?
Yes, now we can.
Yes, great. Yeah. All right. Thank you again for introduction and attendance. My name is Jong Yoon. I'm a PhD candidate at Dawoodi Lab at Purdue University. Today I just want to talk about my pipeline about generating pangenomes using short reads and how we can better utilize that in the context of population and conservation genomics. I would like to make my talk shorter, so if anyone is interested in very deep details, then I would refer you to this paper published in Molecular Ecology Resources. So here's the paper information, so feel free to go and read the paper when you have a time. All right. Yeah, so today I want to introduce the pangenome first, its definition, advantages, and some types, and also introduce my research, the background, and how to generate short-read pangenomes, the pipeline, and the benchmarking results. We compared across different assemblies, so I will show you how they were compared. And I would finish my talk with talking about implications in conservation genomics and the future of pangenomics. Yeah, the pangenome, what's that? So it's briefly, I think it's something beyond a single reference genome. You know, so far with the linear reference genome, we try to represent entire species, their gene pool, with just a single individual derived reference genome. And pangenome is something that overcome the limitations of the linear reference genome by collecting all the variant genes and loci in a focal species into one set. So it's consisting of core genomes here, which is shared by all the individuals in your focal taxon, whichever it is, population species or even genus family. And also there are dispensable or accessory genomes, which are shared by some individuals, but not all. And then unique genomes, which are just present in a certain individual. So you can imagine that dispensable or accessory genomes here and unique genomes might contribute to local adaptation or speciation. Then why we should think about the pangenomes in the context of conservation? Why does that matter? That's because, well, in conservation, we are mostly interested in the general patterns of genetic diversity or inbreeding, how the demographic processes like population decline, population expansion contributed to those factors. And pangenomes can help capture more thorough genetic diversity um, by collecting SNPs and SVs more effectively. So based on that, we can even identify core genetic variants and accessory variants across the SNPs and SVs, and then we can use those information to study adaptation and hybridization and speciation, as I mentioned earlier. And one important thing I think is that using pangenomes, we can avoid the reference bias. As we represent a species' entire gene pool just with a single individual, we often have some missing regions in the species genome. So if we look at this left-hand side panel, there are many reads that could not be aligned to this linear reference genome from the single individual because this individual may miss some regions, likely locally adapted variants. But here on the left-hand side panel, you can see that now there are less unaligned reads because multiple haplotypes are congregated under one single structure of pangenome, and now they can capture more reads, even in the same region. Because of that, pangenomes can increase read mapability, and also that leads to variant calling accuracy and genotyping accuracy, and ultimately that will lead to higher resolution in our downstream analysis as well as their accuracy. Also, pangenomes are better at detecting structural variants and even large genomic rearrangements. Like here, the SVs, due to mobile element variants like TEs, and indels inversions, and even chromosome scale translocations. And I'd like to introduce that pangenomes can be represented in multiple formats. If you have heard about pangenomes, maybe it would be graph pangenomes these days, but not just graph representation. It can also be represented by this linear structure. Um, but this figure here is, I think it's better representing the linear structure of the pangenome. If you look at this number 7, then the pangenome contains all the sequences from the focal species, as long as they are not technical artifacts or contaminants like these purple bars. So here in this number 7, the box, the pangenome, collects all the reference sequences, the red bars, and also even fully unaligned sequences, the navy bars. Also, those mixed ones, partially aligned or partially unaligned sequences, again, as long as they are from the focal species. Graph representation these days actually are better at accommodating complex variants derived from multiple individuals, such as nested SNPs within indels, or as I said, the chromosome scale translocations, because they can represent variants like these bubbles. And bubbles are connected by these edges from node to node. So here in this single structure, we can represent basically, I think, whatever we want as a variant. Like these very complex variant or like these blue lines, everything is available. But the problem is, although it's very optimal, it's computationally very expensive. So it's something we should consider when we use pangenomes in our research. To construct those graph-structured pangenomes, so far the long read is the gold standard because, as you may know, the long read sequencing can sequence through large genomic regions and then repeat regions so that it can capture structural variants very thoroughly. As far as computational resources or your data permits, then you can, I think you can even collect like chromosome scale, like synteny-like structural variants. However, it is very expensive. So for like conservation labs with limited funding, they cannot afford usually. Also, it needs high quality samples for extracting high molecular weight DNAs to do long read sequencing. To compare with long read sequencing, we can think about short read sequencing. As short read sequencing, as you also may know, it has some pros and cons because it can read through only 50 to 300 base pairs. Basically, it cannot read through repeat regions, so that's a huge limitation. But it's very affordable and it does not need any high quality samples. If it can read through just 50 to 300 base pairs, then it's good to go. And also, it does not require high computational resources. So analytically, I think it's more easier and simpler to do than long read sequencing. These comparisons between long read and short read sequencing actually inspired our research because, well, in conservation field, the funding is always very limited. So we should think wisely about how to better utilize the fixed amount of funding to sample collection and sequencing experiments, etc. But we have thought that maybe with short-read sequencing, because we can usually have equal or better accuracy, sequencing accuracy than long-read sequencing, and we can accommodate more samples at the same amount of funding than long-read sequencing, maybe that would be better to capture like species level patterns of genetic diversity or adaptation profiles. Then our thought led to, can we use short-read sequencing for constructing pangenomes? And if that's possible, how those short-read based pangenomes will be different from long-read based pangenomes. So our research started. We first conducted, of course, literature search. And actually, there have been some plant pangenomicists who paved the way for short-read pangenomes. For example, this Yao et al. 2015 paper introduced so-called metagenome-like pangenome approach, where the researchers collected all the reads from all the individuals, like you can imagine something like poolsec, and then they assembled assembly of like contigs from those collected reads all together. So now they have assembly of the study population, and then they reassign those contigs of the assembly to each individual so that they can identify what are the core genomes and what are the accessory genomes. And second, Hu et al. 2020 represented a more practical approach called iterative map then assemble. Here they aligned individuals' reads against the existing linear reference genome first. So they could identify which reads could be mapped to the reference genome and which reads could not be mapped reference genome. So, and they call that as unmapped reads. And using those unmapped reads, they assembled to yield contigs, and they are basically accessory contigs because at least those contigs were not shared with the referenced individual. And appending those accessory contigs to the existing linear reference genome, they updated the reference and making and calling it as a linear pangenome. And those steps one and two were iterated, so that is called iterative mapped and assemble approach. And based on those literature search, we conceived a pipeline shown here. Basically, it's a combination of the metagenome-like approach and iterative mapped and assemble approach with updates by employing modern bioinformatics tools to convert the final linear structure into the graph structure. Just to briefly explain this pipeline, the details can be found in my GitHub repo that I'll show you later. Here in the leftmost column, all the read filtering and adapter removal is conducted. So we just remove adapters and contaminated reads and even ends, and then we pull all the reads together here and move on to the steps in the middle column. Here we first map the pulled reads onto the linear reference genome, the yellow bar, so that we can discriminate which ones are mapped reads, green bars, and which ones are unmapped reads, the pink bars. And separately, between those mapped bars, mapped reads, and unmapped reads, we assemble those independently to contigs here, and again to supercontigs, so contigs of contigs. So we make now these khaki bars, supercontigs from mapped reads, and purple bars, supercontigs from unmapped reads. Then we append those contigs, supercontigs from unmapped reads to the yellow bar, the linear reference genome. And we iterate these whole steps in the middle column again, the second time. Then finally, we update the linear reference genome into this linear pangenome with more appended accessory supercontigs from the unmapped reads. At the same time, we collect all the supercontigs, regardless of whether they are from mapped reads or unmapped reads together. So let's say it's a collection of supercontigs. We have the supercontig pool, then we reassign the supercontigs into each sample. So now we have information of what contigs are belong to which sample. Then using those information, we build the first short-read graph pangenome called MC graph pangenome here. This is called MC pangenome because we use the minigraph cactus pipeline. And also we can generate the second short-read graph pangenome from this linear pangenome using VG toolkit. So it's called VG pangenome, VG graph pangenome. And to do this, we need an extra step to identify the de novo variation within the linear pangenome by realigning the filtered raw short reads onto the linear pangenome. I'll compare the MC graph pangenome and VG graph pangenome also later. So I'm just emphasizing those two are MC pangenome and VG graph pangenome are two short-read graph pangenomes, and this one is short-read based linear pangenome. And here is the brief introduction of the exact procedure we followed to benchmark those different assemblies. So now we could have 5 different genome assemblies. First one, the linear reference genome here, publicly available from the NCBI. And we also downloaded the raw long-read data of the barn swallow from the Sacramento et al. 2023. And from the raw long-read data, we assembled them into 10 haplotypes, and we subsampled short-read data from those long-read-based haplotypes and generated 3 short-read pangenomes that I introduced earlier. And we also generated this long-read graph pangenome following the exactly same procedure of Sacramento et al. 2023, again using the raw long-read data. And here in the right-hand side panel, I want to introduce the different structures of these pangenomes and how they affect read alignment. So in the linear reference genome, some reads are failed to map and filtered out, like the CTA, the purple read. In linear pangenome, these CTA reads can be mapped on the appended accessory contigs from the generated from the unmapped reads. And in the graph pangenome here, even those AAG and ATG reads that were originally mapped to the same genomic location under the same contig are now mapped to separate contigs based on their similarity. So the important point here is that, sorry about that. Important here about that this A site at the middle was variant in the linear pangenome and linear reference genome, but now it is not a variant anymore under the graph pangenome structure because the ATG and AAG reads could found contigs that are more similar to them than just the AAG, the same contig. And in the panel B, I'm showing you the classification of assemblies I used for clarification. Here, linear assembly includes linear reference genome and linear pangenome, while the graph assembly includes VG graph, MC graph, and long read graph pangenome. And the short read pangenome includes linear pangenome, VG graph pangenome, and MC graph pangenome. Now we can dive into the benchmarking procedure. For this step, we downloaded two independent short read datasets from another research group, Shield et al. 2021. The first dataset was consisted of four populations, so we called it as population level dataset. The second one was three subspecies, so we called species level, subspecies level dataset. We compared across the five different assemblies, their assembly structure, length, SNP, SV, and mapping statistics, and variant identity, how similar or different the query variants are against baseline variants. And here we used linear reference genome as a baseline because it's a, traditional norm. And also long read graphene genome as a baseline. I think it will be a new norm in the future. And also we use VG graph pangenome to compare among three short read pangenomes. We also analyzed some population genomics metrics, and we compared like genetic diversity and site frequency spectrum as a summary statistics in population structure, genetic differentiation, and selection scan. Yeah, now we go into the results, and I know it's very complicated, so I'd like to bring your attention to here first. The size compared to the linear reference genome, well, it's basically increasing with the complexity of structure. So the most complicated structure in long read graphene genome, it has 12.2% larger size than the linear reference genome. And moving on to here, the genetic diversity matrix, I'm showing you the nucleotide diversity and F-raw, which is the inbreeding coefficient at the whole genome level. I'd say it's largely similar across five assemblies, although the F-raw value is lower in linear assemblies here, and that's also because of the effect of structures. I'll explain it later. And as we count, I'm highlighting the post-filtering number of variants here. So basically, the graph assemblies, the VG, MC, and long read graph pangenome here has the larger number of variants. And also because of, thanks to the graph structure, they could use another algorithm for structure variant calling. So using VG in this study, they could capture more large scale structural variants as well. And variant identity results, I'd say in general, short read pangenomes could capture the most variants of the baseline assemblies. For example, when we use the linear reference genome, both SNPs and SVs in the linear pangenome were almost just identical. And when we used long-read graph pangenome as the baseline, both short-read graph pangenomes, the MC-graph and VG-graph pangenome, had reasonably similar SNPs and SVs, although the MC-graph pangenome had more and similar larger-scale SVs than VG-graph pangenome. And when we used VG-graph pangenome as the baseline and compared across the three different short-read pangenomes, a more similar variant sets were found between graph pangenomes, the MC and VG graph pangenome, than between a graph pangenome and a linear pangenome. So we could see that there must be some effect from the structural difference of the assemblies. And here are the results of population structure and site frequency spectra. So this is the SNP-based result within the same subspecies, the population level dataset. And you can see that they are almost just the same. And I'm not showing here, but the linear reference genome also showed exactly same result. And moving on to the SV-based result from the subspecies level dataset, again, we could see that the population structure and summary statistics were captured almost the same across the different assemblies, including even the linear reference genome. And the genetic differentiation and selection scan results, well, I'll not talk about the details here, but the general pattern showed us that although the FST values, the genetic differentiation values, were higher in graph assemblies, the patterns of population differentiation were found the same regardless of assembly types. In other words, the rank order of population pairs based on their genetic differentiation values, they were just the same across the assembly types. And in selection scans, more selection outliers were found in linear assemblies, the linear reference genome, and linear pangenome. And we think that's because, again, the structural effect. So just to remind, in this graph pangenomes assembly, there should be lower number of variants because in this graph structure, reads can now find contigs, different contigs that they can align to. Originally in linear assemblies here, they could capture it under the same contig at the same genomic location, so they were identified as variants, but not anymore in graph pangenomes. So that will lead to the lower number of variants, SNPs in this case, and that also again lead to the lower number of selection scans and also lower values of FST and FRAW, the inbreeding coefficient, because those variants will break down the tracts of homozygosity when we use in calculating the FRAW values. So now we can talk about discussion. Some key finding is that we found the short-read pangenomes could be used as alternative to costly and complex long-read approaches. And it is equally powerful and even more accessible for conservation genomics. As I showed, I think the short-read pangenomes effectively capture genetic variation resembling either the linear reference genome or the long-read graph pangenome, and the population scale analysis were reliable. And it's of course more economical and accessible than long-read approaches. So we believe it can lead to democratization of genomic research across endangered species. Because when we sample endangered species, it is very difficult to collect high quality samples. It's usually degraded and usually invasive, non-invasive. So we need to sometimes extract DNAs from fecal samples. And it also helps research in resource-limited regions, of course. And thanks to the graph structure in short-read graph pangenomes, it had comparable read mapping metrics and accuracy. And the graph assemblies also can detect more SVs. So that will help SV-based research in the future if you need to use the short reads. We also made some practical recommendations of using short-read pangenomes in our paper. Just to briefly talk about, if your research is focused on SNPs more than S3s, then linear pangenome might be ideal because it can offer computationally efficient analysis than graph assemblies. But if your research is more focused on S3s, then graph pangenome might be more valuable. Well, in comparing and deciding between MC graph pangenome and VG graph pangenome, if you decided to go with some short read graph pangenome, then you should think about the trade-off between computational resources and workflow because MC graph pangenome requires higher computational resources, while VG graph pangenome includes an extra step to identify de novo variation when it needs to be converted from the linear structure into graph structure. Lastly, if you need high resolution structure variants and if you need to analyze repeat regions, then you should go with long read graph pangenome. It's inevitable because short read graph pangenome anyway cannot read through and analyze the repeat regions contiguously. There are, of course, limitations in reality. Well, as a general challenge, well, all the short read, not short read, all the pangenomics work require high memory use and insensitive processes. Intensive, it's a typo here. Time intensive processes. Um, and there are active developments in software. So, well, it's very frustrating, but depending on the software versions, results will be different. That happened to our case as well when we generated the long-read graph pangenome using the same process of the original authors, using the same data of them as well. Yeah, that's kind of frustrating, but it's reality. And furthermore, most pangenome tools are optimized for model species, but so there should be more testing using non-model species data. And variant uncertainty, I think, is the most frustrating part. Because, well, even if the overall variant identity was matching in our case across different assemblies, the exact variant profiles will be different. In other words, the set of variants you will find from the linear reference genome will be different from a short-read pangenome or even from a long-read graph pangenome. And we have no way to identify which variant set is a biological truth or just technical artifact. And one last note, as I mentioned earlier, the linear reference genome also showed exactly the same results with the other pangenomes. We believe the previous studies, based on the single reference genome so far, should still be informative for conservation. Yeah, so then what's the key impact for conservation genomics? Well, we found that our researchers can utilize limited funding to fully characterize gene pools, especially structural variants, and it will be better for conservation strategies. For example, in genetic rescue programs, we are interested in to find the best donor population for recipient population, and the donor population's adaptation profile should not be too different from the recipient population, because in that case, the donor population's genome will override the recipient population, which can lead to outbreeding depression, and the donor population's adaptation profile is not adapted to the habitat conditions of the recipient population, so it will ultimately lead to reduced fitness after the genetic rescue program. And we believe the better capturing of structural variants and identifying adaptation profiles based on those structural variants will help mitigate that outcome. And I'd like to wrap up with talking about the future of pangenomics. Well, it's not just my opinion at all, but just a broad sense opinion. The community is now believes that pangenomes will likely be a new standard reference, especially for studies using structural variation. because, well, pangenome, especially graph pangenomes, can accommodate structure variation very well thanks to the structure, graph structure. And spore pangenomes will likely emerge soon. And I think it's actually emerging now in the non-model species field as well. That can mitigate some biases arising from the use of single species-based reference genome. For example, in hybridization studies. In that cases, you want to include variation from both species of interest to avoid any bias from using just a single species reference genome. And not just pangenomes, but also pan-transcriptomes and pan-epigenomes are emerging now, actually. Then advancing computational resources will and should make using pangenomes easier in the near future. Therefore, we need a standardized best practices of doing pangenomics is needed as soon as possible. With that, I'm showing you here my GitHub repo where the scripts are available. And here are some key points of my pangenome pipeline, but I don't think my pipeline is the gold standard. But if as long as some key points like the metagenome-like approach or um, iterative mapped and assemble approach are combined, and also if you can combine some approach to convert linear structure to graph structure, then that should be viable as well. With that, I want to thank to my co-authors, Andrew Black, Natalie Allen, and my advisor, Andrew Doody. And the project could not be done without huge technical support from Purdue RCAC and Dr. Glenn Hickey here, who's the software developer of VG Toolkit and Minigraph Cactus Pipeline. He even updated the softwares for us. And I'm glad to have huge analytical advice from Dr. Bohel Fang, who's working actively on non-model pangenomics. And also thank for earlier feedback for the research from Duty Lab members, John Bickham, Andrew Mularo, and Julia Buchanan-Schumanke, and my advisory committee members, Drs. Bonnie Downing, Jessica Gravich, and Nina Darkelson. If you are interested in learning more about pangenomics, then I'd like to direct you to these papers, which are very readable and very informative review papers and a dissertation. I strongly recommend reading those papers. With that, thank you for your attention, and I'd like to open the floor for any questions you have.
Thank you so much. What a great talk. We will take questions. You're welcome to write them in the chat if you would prefer, or you're welcome to put your little virtual hand up and then ask your question out loud. And I can start us off if other people are still thinking a little. So you talked about some of the challenges,
Mhm.
technical, financial, in getting high quality, like high molecular weight DNA in the field or from species where you're worried about uh, like the invasiveness of sampling and things like that. Um, so I think it's really helpful to see like the use of short reads in a way for conservation genomics in this pangenome approach. You're still starting with a reference genome. So I'm wondering, can you speak a little bit about do you see there being a way forward if you are working on a species of conservation concern that still doesn't have a reference genome and you only have short read funding? Do you
Mhm.
think there's an approach there with from
Mhm.
pangenomics, or do you think it makes more sense to still try to put together a single individual reference?
Yeah, that's a really great question. I think there could be a couple of or a few different ways to avoid that limitation. One would be using closely related sister species reference genome. That might be the easiest way. The other way would be just assemble a de novo reference genome using whatever data you have available. Could be short read data. Because theoretically, pangenome is just, well, as I mentioned, it's just an entire set of sequences of the species. So as long as you can build a de novo reference genome to use as a backbone when you build the pangenome based on that, then it would be fine, theoretically, I think. Or there's another approach of building pangenomes. I used so-called backbone-based pangenome construction approach here, but there's another approach called a backbone-free approach, whereby you can just um, well, I'm just saying in a best case scenario, you have context, high quality context, probably based on long reads, and just across those long read contexts, you cluster them based on their similarity. So just you make communities of those clusters, and that will make something like I say cluster pangenome or community pangenome. But that last approach might be more difficult using short reads because we cannot make high quality context anyway. Um, yeah, so there are some approaches in short. I would recommend finding closely related sister species reference genome.
Thank you. And we do have a question in the chat. It says, do you want to ask it yourself?
No, please go ahead. I was
Okay.
going to ask a question, then I saw this.
Um, awesome. So the question is, what should the minimum coverage be?
Mhm.
Would 30 times coverage be good enough?
Yeah, I think 30x, yeah, should be well enough. Yeah, the coverage will matter when you assemble those short reads into context. So you want to have higher depth as high as possible to make high quality, very robust context, even before generating those contexts. Here in this case, because we pulled all the reads together before um, assembling context, let me say the total coverage it was 200x. And actually, the meta-genome-like approach by Yao et al. 2015 was to avoid the low coverage issue. So in their case, they pulled all the reads together and assembled context because they had like even less than 1x coverage at each read. Um, Yeah, I think that will mitigate some issues, but my recommendation would be um, yeah, 10x per sample per individual.
Yeah, I'll ask something related to that question first before I ask my question. So 30x coverage is per sample. Like, how many samples would you need ideally to capture like a good amount of pangenomics?
Yeah,
Cost effective and yeah.
that's another great question. So in conservation, we are more interested in species-wide patterns. So, well, it's just my personal opinion, but I think it's more important to accommodate more samples than higher depths of coverage. So that's always kind of tricky part to plan the sequencing and sample collection. But I would say um, at least, well, 30 samples or more would be best across the species range. And also, of course, that should be species specific, depending on how large the species range is and how complex their genome structure is. Yeah, but I would say at least 30 samples, just thinking about in terms of statistics. Yeah, and then I would determine the depth of coverage after fixing the number of samples first, if that answers your question.
I think so. Yeah, I guess it'll also have something to do with the amount of genetic diversity, right? Like if the genetic diversity is really like cheetah or something that have very limited genetic diversity,
Right,
then you probably aren't going to gain anything with the pangenomics
right, yeah,
compared to the linear reference genome, right? So
right, yeah,
I guess the number of samples would be like you said, would number of factors involved
hmm
in that to decide. So I think 30x,
hmm
I mean, this coverage per sample and the number of samples all depends on the species in question, I guess. X.
yeah, yeah, and more practically funding.
Of course.
Yeah.
If my question was something related to like the short read genomes, do you really see a niche for short read like pangenomes with the advent of new tools like minigraph and all, like Cactus, especially the long read becoming cheaper and cheaper?
Mhm.
Will there be the short read pangenomes niche like it going forward?
Yeah, well, it's ironical to say like this, but I hope not. Because I hope long read sequencing becomes cheaper, faster, so that even every conservation researcher can take advantage of long read sequencing than using this short read based pangenome approach. But just the reason I thought about this was basically um, well, I know that most conservation labs are not able to afford the cost of long-read sequencing. So I think there would be some niche for a while, but I hope that would ultimately go away in the near future.
I think a lot has to do with the like how mainstream the long read becomes, right? Like it's still
Yeah.
not mainstream. Many universities still doesn't have like the core facilities equipped with long reads and everything. So I
Mhm.
guess there's a lot of factors involved there as well. Thank
Right.
you for the talk. I think there's another question um, in the chat.
Yeah.
Ellie, do you want to go ahead?
Yeah. So the next question in the chat is, if I
Mm.
have a collection of reference genomes, what's the best way to then create a pangenome? One thing I did a few years back was to identify similar sets of genes and use something like MC-scan to identify collinear regions. But I'm wondering if there's a better way to
Ah.
go about it.
Yeah, so it's a good question. So, well, first of all, I'm not familiar with the MC scan by myself, but these days there are advanced pangenome construction approach. You can you can find some resources from Fuji Toolkit, or Cactus, which has a pipeline called Minigraph Cactus pipeline that I used, or just Minigraph. And if you are just working exclusively with the reference genomes, um, then Cactus has another pangenome construction approach, progressive Cactus, that use genome assemblies of different species and based on the phylogeny of those species. And also PGGB, which will be worth to look at. It's the backbone-free approach that I mentioned earlier. It can cluster contigs within the reference genomes based on their similarity, sequence similarity, and make a community of them. So, and you can do like making communities per chromosome or per contig. Um, yeah, there are some advanced methods these days. Does that answer your question? Harisha? Harish?
It does, thank you.
I can add a little bit. I work with maze genomes, like with a lot of repeats, TAs, and huge genome size. I've used PHG in the past. I think Jong Yoon mentioned about it, but yeah, I highly recommend it.
Mm-hmm.
You can provide it. It's from the Ed Buckler's lab from Cornell.
And I've helped support some of the Cactus tools on our cluster as well. And they can be, depends a little bit on what you're putting into them, but they can be quite like memory and time heavy, like Jung Yoon did mention. But I only heard like recommendations and sort of appreciation from the researchers using them at the end. So they definitely are possible to make work on a cluster system. And folks like us are happy to help make that happen too. I think we have time for another maybe question or two if anyone else has anything on their mind. Awesome. Well, we I think have covered quite a few topics off of your talk. I really enjoyed it. I hope everyone else did as well. So let's thank you one more time for speaking to us today, and happy Friday, everyone.
Thank you very much.